Hey, Morris.
Hello, Lance.
Good to see you here.
Good to see you here.
Yeah, so last time here, I had a discussion on the HUGO models in general with KG.
I have seen that.
Yeah,
fantastic.
Yeah. Today we're going to focus on the neural diseases.
Sure.
And talk a little bit about yourself. You're a neurologist by training.
I'm a neurologist by training. I was a practical neurologist.
Yes.
And after
where did you practice?
In the Russian Federation, St. Petersburg.
St. Petersburg?
Yes, St. Petersburg.
So you were a medical doctor?
I was a medical doctor.
In
the hospital.
In the hospital?
In the neurological department, yes.
Okay, so you were seeing patients?
I have been seeing patients for 3 years.
Okay.
Yes, every day.
In the Russian language?
Yes, in the Russian language.
Wow.
Occasionally with English, of course.
Being a doctor in Russia, seeing patients in the Russian language.
English and other European languages.
Wow,
of course.
that's amazing.
Anyway, after the practicing part, after being a neurologist, seeing patients every day, I decided to dig deeper in the scientific aspects. So I started my PhD training in Germany, more specifically in Ludwig Maximilian University of Munich. There I studied neuroscience, particularly in neurodegenerative diseases, with Professor Johann Hams for around 5 years. And I also get my PhD there in neuroscience.
Okay, so yes, you've been with Cyagen for a year now?
I've been with Cyagen for years, more than a year.
More than a year?
Yes.
Time flies.
Time flies. Temper fidget, always.
Good. Now you're in the science part of it, but I guess you always have that kind of human touch because you've seen so many patients.
That's very interesting for me because my clinical practice experience gives me certain aspects from the patient side,
yes,
from the clinical side.
Yes.
And also after that, I've been in academia for 5 years, and now I'm on the industrial side.
Okay, so today we're going to talk about Alzheimer's disease, AD, and the mouse models.
Sure.
Yeah, first of all, AD probably is the biggest human disease.
Yes.
Most prevalent globally. Affecting over 50 million people. And
it's growing.
And it's growing
year by year.
With the aging population.
Yes.
Yeah. And still, this is a huge unmet medical need. Because as we know, well over 99% of the clinical trials on AD ended up in failure. And a handful of drugs that were approved only have very marginal clinical benefits.
Yes. As we understand, the most advanced antibody treatment for AD, donanemab, lecanemab, based on the phase 3 and also the following clinical trials and also the therapeutic studies, let's just say the benefit of taking these drugs for, let's just 3-4 years, the overall scoring benefit is around 1.1. I think it is out of 20 something or 30. So it's not visible, it's not predominant, but it's firstly statistical significant, secondly it's something. Over 100 years after Dr. Alois Alzheimer discovered Alzheimer's disease in his patient, we have been struggling of getting the bottom of this disease, and after 100 years, this is what we get. This is not much.
Yeah. Well, you can say that AD probably is the Waterloo for all the drug development companies so far.
Yes.
So many failures, so much money spent, but this is something that we cannot give up on.
You cannot give up on such a population, and it's globally speaking, you cannot give up on such a patient population. And for individuals, you cannot give up on your family.
Yeah, of course.
Yes.
Of course. So now let's come back to the mouse models. We know there are some of the very popular AD disease models. Some of them are from the Jackson lab. I looked at quite a few of them. Looks like they all have their own limitations. From these two famous A-beta pathology and the Tauopathy. I guess previously a lot of people were focusing on the A-beta pathology, yes. Right? Getting rid of the plaques. But looks like it's not the root cause of the disease. And people are shifting focus to tau, which we're going to talk about today.
For many years. First, about A-beta and tau. It was detected by Alois Alzheimer 100 years ago. In the first site, when he looked into the slices of his first AD patients, Augustus Detail. One thing interesting is in the lab I was working, we still have some slides from Augustus Detail.
Wow, you were from that lab?
From that lab.
Oh my goodness.
Wow, it's something that good. I from the lab where we first discovered AD. It's also the fact we still didn't solve the problems.
We're still in the darkness.
Yes.
Trying to figure out.
Anyway, the two hallmark A-beta plaque and tau was detected 100 years ago. And for the following 100 years, there was millions of debate. Which one is what? Which one is the first? It's like the problem of X and him. You first get X, or you first get him. So, and for the last 10 years, as you just described, so much money invested. And around 5 or 6 years ago, I believe, the first really working, debatably really working antibody drug for amyloid plaque, the aducanumab, came out. And that's the symbol of the entire A-beta target treatment getting back to our side. And it gets growing, it gets hotter in the following years, and now is the predominant way of treating Alzheimer's disease. So the understanding of the A-beta as the primary pathological hallmark is not purely scientific thing. It's a scientific, commercial, and lucky things.
Okay.
Aducanumab worked firstly. So it puts our side in A-beta as its primary target for AD. It does not undermine the importance of tau. However, even from a scientific aspect, if I have to compare A-beta and tau, tau is a really difficult target.
It's really difficult, but probably it's more closer to the root cause of the disease. We don't know.
We don't know. And as a previous scientific worker, scientific researcher, whose main focus was A-beta, I also have to say that, yes, based on numerous literature review, we understand that tau is so tightly connected to the pathophysiology of AD. It's so tightly connected to the most important thing in neurodegenerative diseases, which is actually the death of neuron.
Yeah.
It's directly caused the death of neuron.
Not just AD, but FTD and some other diseases.
And other diseases. It directly reflects neuronal death. And it directly reflects the cognitive impairment along the entire disease pathophysiology.
Yes.
But still, it's hard to learn. It's hard to study.
Well, it's hard, you know. The field is littered with failures, but we're still working very hard. And we hope that one day we're going to hit something.
Actually, one thing makes studying tau so difficult is compared to the numerous AD mouse models, the A-beta mouse models, we sort of say that the APPPS1, the 5XFAD,
Yes.
APPPS1 delta E9,
Yes.
and the APP whatever thing.
You name them.
We have very few tau models. And very few tau models, they don't always work as expected. That makes tau very difficult to study from the beginning of animal models.
So for the past a year, you've been working with the tau model.
Yes, for the past
Inside.
year, if the past entire year, the 12 months, I've been working with a newly developed mouse model at Cyagen.
Tell us
The
about
humanized tau model.
it. Or the HUGO-Tau.
Yes, the HUGO-Tau. Well, before talking about the HUGO-Tau as a new word, the HUGO,
Yeah. Yes.
I would like to talk a little bit about the entire background of the HUGO project at Cyagen.
Yes.
So, I believe you have already talked quite much with KG the last time during your webinar.
Yes.
About the humanized project. I would like to emphasize again why it's so important. The HUGO is short for Humanized Genomic Autolog.
Yes.
That actually means replacing the entire gene in the mouse,
The genomic sequence.
genomic sequence in
Not
mouse
just the CDS. Yes.
by human, by human genomic sequence.
That's right.
Yes. As I understand, at least during the last year, that we at Cyagen has a technical advantage. Using our TurboKnockout technology, we actually can replace the entire mouse genomic sequence in the ES cell for a sequence as long as up to, I think, 350 kb.
That's right.
Yes, that's
on our
much.
record is 400 kb in one round of targeting.
That's already something incomparable for the most popular CRISPR-Cas9 technology, which is, yes.
Well, with CRISPR, you know, if you're talking about a knock-in or endogenous locus, the upper limit is roughly 20K. It's very hard to go beyond that limit.
And now we have hundreds.
Now we have hundreds.
But one thing actually I'm curious, I was, at least based on my training, I understand that the ES cell targeting technology, it requires a long time to make a mouse model.
Well, you know, it's still the golden standard of, you know, genetic modification in mice. But when CRISPR came out over 10 years ago, people all rushed to using CRISPR because, you know, it's easy, it's cheap, you know, everybody can do it.
It's fast, it's cheap.
Yeah. But with ES targeting, you know, it's like a very complicated procedure. It's like a surgery, right? It's not everybody can do it easily in their labs. So I remember, like, about a dozen years ago when people started to using CRISPR, lots of our competitors would say, hey, yes, targeting is obsolete. Let's forget about it.
But we are still doing that.
But we're still doing that. We're still making improvements upon improvements. And to the extent that we can do very complicated modifications of mouse genes, you know, up to several hundred kb at any given time, and in multiple locations. And one round of targeting, we have squeezed it down to like around 6 months, which is not too much different from the CRISPR project.
Well, as far as I understand before, I mean, without that Cyagen technique, the usual ES targeting takes years.
Oh, it takes at least 12 months.
Yes, and if you want to purify, if you want to, let's just say, modify the strain that you get from the first round, it takes another 12 months.
Oh yeah.
Yes,
Easily.
that means a long time.
Yeah.
But one thing I'm quite excited when I joined the Cyagen is I was taught that the TurboKnockout, which is a Cyagen patented technology, can squeeze the entire one round into 6 months, and that's actually
right.
a really accelerated process.
Yes, yeah, that's
It's
right.
fast.
That's right.
Well, that's something that is really exciting because now we have long sequence, short time, and put it together. That means almost a much purified, much, let's just say, modified technology of getting this new mouse strains. Yes, and Tau, what we just talked about, the humanized Tau model, is made by this technology.
Yes, yes.
Yes.
Among, another 100 some.
Among another
HUGO
100
projects.
something which we are going to talk about.
That's right. That's
Yes.
right. Yeah.
To make this HUGO strain, or the significance of HUGO strain, is we replace mouse genomic sequence by human, as I just described. And the purpose of doing that is to make the mouse model human relevant.
That's right.
So we replace this gene, then we expect to get a humanized process. We expect to get a humanized process related to this gene in the mouse.
Yes, the gene is in the right context, roughly, right? And, the expression, the regulation, and even the splicing, because sometimes you have alternative splicing, right? Without the whole genomic sequence with the UTRs, introns, exons, there's no way that you can have a faithful splicing mechanism still in the mouse. So, yeah, we have already seen lots of benefits of these models. And the HUGO concept that we came up with now is gaining traction. People are coming to us, hey, Cyagen, can you make a HUGO this, a HUGO that? So this is a concept that beginning to get popular.
It's not just to get popular. It's not the right phrase. It's not popular. It's relevant.
Relevant, yes, clinical relevancy.
It's relevant. Why it's relevant? It's also why I was so enthusiastic about studying these models at Cyagen. When you replace a mouse gene by human gene, partially replace this process. And when you have the humanized process in the mouse, especially when this process is disease-relevant process, you may just get the relevant diagnostic technique and the relevant treatment also applicable in this mouse. Whatever you would like to try to develop for human disease, now you can use this mouse model because the human gene is there. The humanized process is there. We call this kind of study the translational study. And where we to translate it to human.
That's right.
That's why I'm saying this is not just popular. It's of course popular, but the reason, the root for this popularity is relevant.
Okay, so let's delve into it a little bit. So you've been studying this model for over a year, and so in your hand, what do you see? I mean, we have the wild type humanization of the Tau, human Tau, right? And we have the on top of the wild type humanization, we have the put in the mutation, the P310 mutation.
Yes.
Right. So what have you been seeing so far?
As you just mentioned correctly, we make 3 models actually out of Tau.
Okay.
One is humanized Tau, wild type. Another one is humanized Tau with the P301S mutation. And the last one is P301L mutation.
Yeah, the two famous mutations.
The reason we do this is when we replace the entire human gene into a mouse model, technically, we created another species.
Another species?
Yes.
Or a chimera, so to speak.
Yes, a chimera, which is not human entirely, of course, but not mouse entirely. That means if we would like to use our humanized model to study any disease, we have to have the correct control. That's
right.
another thing I think, and I insist we do that to make the entire study sophisticated and reliable. We have the correct control. We have the humanized tau wild type.
Yes.
Yes. And based on this, we have tau P31S, P31L. Before talking about what I have seen in this mouse model, I think you're very interested in that. Everyone's very interested in this kind of thing. I'm afraid I have to be a little bit tedious. I have to first talk about why we need humanized tau. Not just because you mentioned that in Alzheimer's disease, tau is 50% the most important pathological hallmark. Tau represents half of the AD pathology, and tau represents the major pathology in a bunch of other diseases, such as the frontotemporal lobe degeneration.
Yes.
It's because in the root, the human tau and the mouse tau, they are different.
They're different.
They are completely different things.
How?
Tau, or we call it microtubule-associated protein tau, is a protein inside mostly neurons. It's like the scaffold to make the neuron itself stable and functional.
Okay, so it's a structural protein.
Yes, it's a structural protein. And as many other this kind of protein, it has isoforms.
Isoforms?
Yes, isoforms, which means one sequence may make different proteins. They are similar but different. There are many different isoforms for tau, but just for the convenience of classification, we use two marker parts to classify them. One, we call it the N-terminal inserts, or in short, N. Another is the microtubule binding repeat. We call it R. Yes.
Okay.
The interesting thing is, there are three types of N. You have either 0N, 1N, or 2N. That means you have 3N.
Okay.
And there is two types of R. You have either 3R or you have either 4R.
So 3 times 2, you have 6 isoforms.
Yes, you have at least 6 isoforms. And there are many other isoforms.
What about mice?
What about mice? Good question. That is the $1 million question. In adult mouse, there is only 4R. That means we have only half of the tau.
Okay.
So if you would like to use, let's say, transgenic mouse model, which you put point mutation inside the tau sequence, the murine tau sequence, what do you get? You get 4R tau and 4R tau related mutations, right?
Okay.
Are there 4R tau associated human disease? Yes, we have that. We have the progressive supranuclear palsy, we have the cortical base degeneration, we also have the atrophic grain disease. We have 4R tau related disease.
Yeah.
But the problem is, we also have 3R tau related disease.
Okay.
Such as pig disease. And to make it even more disastrous, we have 3R, 4R tau related disease, which is a complete mixture. And Alzheimer's disease you just mentioned is a mixture.
So that's one of the reasons that we need the HUGO.
That's one of the reasons why at the very beginning I insisted that for studying tau-related disease, we have to put human tau inside mouse. Otherwise, we lose half of the diseases. We lose half of pathologies.
Okay, so this way have better human relevance.
Yes. This is one of the key reasons why this HUGO-Tau is important for studying human disease.
Okay.
Human tau is there.
So what do you see in these models?
Well, the disease we are going to study is related to tau. As I just mentioned, 4R, 3R, 4R, 3R mixture. The key thing of this degeneration, the Alzheimer's disease, is what we call the cognitive impairment. Or in a more plain language, it's a deficit in learning and memory. So we put together experiments, includes almost 100 tau mice from those 3 lines, and also we included C57 black 6, or we just call it black 6.
Yes.
Just do that as a control. So in total, around 100 mice for an experiment which lasted for 9 months.
9 months?
Yes.
Wow.
This is important because, you know, for human, the neurodegenerative disease, the Alzheimer's disease, FTD, whatever things, it does not happen in younger people. God bless it. It does not happen to young people. It happens to senile population. So for studying this kind of thing, we cannot accelerate. We cannot skip this process. We have to wait. For this, I truly thank the animal department at Cyagen. They bared this experiment with me for 9 months.
Which is not easy.
Which is completely not easy. We have conducted hundreds of rounds of behavior tests and all that other things.
Wow.
Let me just explain our experiments. We set 3 time points, at 3 months old, at 6 months old, and at 9 months old. So we focused on cognitive aspects, and we also focused on other general vital aspects of the mouse, such as the motor function, the anxiety, and other general behaviors.
Yes.
To put it in a more academic way, we set up several behavior tests, which includes the novel object recognition test for learning and memory, open field for general behavior, and the rotor road for motor functions. Okay.
So beside the behavior studies.
After that, after 9 months of tedious work, hard work, we conducted pathological studies.
Yes.
Using both staining and Western blot to see how the proteins expressed in the brain. But that's something we are going to talk about a little bit later. You need to have some patience. We have been working on this for 9 months.
Now we're talking about it for what, an hour? So
okay. So at the very beginning, we conducted the open field test and also the rotor road to see the general behavior in the mouse. As expected, no alterations. For this no alterations, I have to explain that means the entire HUGO series, the 3 HUGO line, they do not deviate from the black 6. And also, within the 3 HUGO line, the mutant lines, they do not deviate from the wild type line. That means at 3 months of old, it's safely for us to say there is no behavioral pathology. The next time point is 6 months old.
6 months.
Now things are getting interesting. We didn't see too much of the motor dysfunction in those lines, neither compared to their own HUGO wild type or the black 6. But we did see some deficit in cognitive function, especially in the P301S mutation line.
Okay.
In the novel object recognition, we see that the H-tau P301S mutant mice, they didn't recognize the novel object in compare to the old object they have been playing with for, I think, 24 hours. Well, you know, the mouse, they like new things. They like new things. They like exploring new things. So if it can recognize the new things, they will play with that for a longer time. But that didn't happen with the 301S mouse. That means they didn't remember what they have seen.
Cognitive deficit.
That is interesting. That is even more interesting. We didn't see anything in the P301L line at 6 months old.
Okay, maybe it's too early for it?
Yes, it's too early. And this is exactly happened in human patients.
Okay.
The human patients of FTD, the human patients with AD who bears the P301S mutation
has an early onset.
Yes, has early onset and accelerated pathophysiology.
Okay.
That means at least in terms of the novel object recognition test, in terms of this episodic memory test, our HUGO mouse behave exactly like human. By the end of our experiments, 9 months old time point, we still didn't see any motor dysfunction in all those lines, but we did see the cognitive deficit in both P31L and P31S. Now it's catching up. The P31L is catching up. Eventually, the cognitive deficit happened as expected, yet slower than P31S. Also exactly like in human.
What about the wild type tau?
The wild type staying functional, clever, capable of recognizing the new things.
Not too much
which
different
is very
from...
important, actually, because despite all our hope, we expect the H-tau functions in mouse as in human. They still human sequence.
Yes.
So there is a slight possibility that this wild type human tau doesn't work in mouse.
Sometimes.
Now, sometimes. It's unfortunate, it's unwanted, it happens.
But not this time.
Not this time.
Luckily.
By comparing the HUGO-Tau wild type to black 6 mouse, we didn't see any alterations. Not in cognition, not in general behavior, not in motor function.
Wow.
This time, it worked.
Wow. This is beautiful.
This is just the behavior side of the H-tau series. By completion of the behavior set, thank you again, animal department. By completing the animal behavior test, we conducted pathological test using the brain tissue from all those 4 strains, wild type, HUGO wild type, P31L, and P31S. While in human patients, the most important pathological hallmark by staining is what we called neurofibrillary tangle, which is a tau aggregation inside the neurons.
Yes.
So this time we expect to see the same thing in those mouse models. For this experiment, we designed 2 arms of the experiment. One arm, of course, the staining is the major part.
Yes.
Using 2 types of antibodies. One is the HT7 from Thermo Fisher, I think. The HT7, which is one of the gold standard for tau detection.
Yes.
It detects only human tau, not mouse tau. Another is 88, detects only hyperphosphorylated tau, which is the pathological related tau, and is majorly human. For another arm of our experiment, we designed Western blotting, which is mainly for differentiating the wild type mouse and the humanized mouse to see, well, at the genetic level, we validated that the human tau expressed only in the HUGO and the mouse tau expressed only in mouse, but we still want to see at the protein level if it is really the thing. So for this purpose, we use another antibody we call Tau5 as a control. Tau5 detects both murai tau and human tau.
Okay.
So with this sound complicated design of study, let's see the result. This is the staining result. And as expected, using the HT7 antibody, we detected nothing in the black 6 mouse.
Of course.
Of course, it's expected there is no human tau. And also as expected, we detected a bunch of tau in the H-tau wild type. That means the human tau is expressed as expected.
Yes.
And interestingly, we detected also HT7 signal in the P31L and P31S mutant lines.
Both mutant.
Yes. But the interesting thing is, the pattern of tau expression is different. Here you can see many of these tau aggregations in the neurons.
Yes.
By the way, I didn't explain, this is the hippocampus of the mouse.
Okay.
Hippocampus, the central of memory.
Yes.
Is our behavior test related. We see this aggregation in the P31L and the P31S, indicating there is a tau aggregation happening inside those neurons. this is part of the validation. With this HT7 antibodies, we conducted our Western blotting. And I think this result is self-explanatory. Yes, you see the HT7, there was nothing in the black 6. However, there is a bunch of things in our HUGO series. And by using Tau5, we also see in all of these strains, the Tau was detected. However, interestingly, in different patterns. That means the Tau isoforms, the Tau bands in the HUGO series differentiate from the black 6. Also, another evidence corroborating that we have human Tau inside those strains.
Yes.
Not just the murine Tau.
Yes, and all the human isoforms.
Yes, and all the human isoforms, which is such a beautiful thing in those HUGO strains.
Wow.
And after the HT7 detection, of course, we ought to detect the pathological Tau. We ought to use the 88 to detect it. And as expected, we didn't see much in the black 6.
As expected.
As expected. And this time, we didn't see much also in the human Tau wild type. As expected, there is no pathology. They behave normally, just like the black 6.
Yeah.
And we detected a bunch of signal in the P31L and the P31S. Since 88 is specially detecting the phosphorylated Tau, we could safely say that there are a severe hyperphosphorylation process of Tau happening in the brains, especially in the hippocampus of those mouse.
Which is characteristic of the AD.
Of the AD, of the FTD, of any Tau-related pathology.
Okay.
That completes our experiment. I think I might just oversimplify the experiments by delineate all the results in a such easy way, but still, it's 9 months. Yes.
Wow.
We gathered tons of data from those mouse, and we now have very solid data saying that the HUGO series, the HUGO mouse models successfully simulate human Tau-related disease.
Well, you know, even before you conducted all this 9 months research, we have already shipped tons of these models worldwide. So people love to use this model. I guess for a reason now I start to understand.
Well, I'm quite sure that all our customers who get this mouse even before me, they saw the same thing as we have been seeing for the last 9 months. And yes, you're right. There is a reason why people love to use
I didn't know.
this mouse.
I know why people are asking us to ship hundreds of these animals to them, and sometimes they only need the wild-type humanization. I was wondering why, because we had the wild-type first, right?
Yes.
And the mutants.
Yes.
Even before we had a mutant, people were asking for the wild-type. So
even a partial, let's just say part of the series, we have 3 models at all, right?
yeah.
And we have the wild type firstly. Even with only wild type, this mouse model has a significant meaning for both the Tau-related therapeutic strategy and diagnosis strategy.
Yes, yes.
For the last several years, as we understand that the majority of AD therapeutic trials focused on A beta side. But that does not mean we forget about Tau.
Yeah.
Actually, there are many diagnosis and also treatment trials going on about Tau. As I show you here, this is a Tau PET-CT. And you can see that for patients with different Tau-related disease, the PET show that the Tau distribution in the patient's brain, this is all human, are very different. That's one thing we say that Tau is important for diagnosis of neurodegenerative diseases because its pattern is distinguishable, is clear, and related to disease. There is another evidence, or another supporting evidence for this thing, is here. This is another literature. When we compare the PET-CT, this is AD patients, by the way. When we compare the PET-CT for targeting A beta and PET-CT targeting tau, it's very easy for you to see that the hotspot for tau PET is much more distinguishable and much more clear in comparison to the A beta.
Of course.
Yes. That means tau can provide a clearer, a more accurate result based on only PET-CT, which by itself is not so accurate because it's just imaging. It's not pathology. This trend also extended to the behavior side. I'm not sure about how you understand the diagnosis of AD and also the other neurodegenerative diseases.
Not much.
But I'm sure you are aware that in recent years, the scientific and also medical communities, they apply more and more non-invasive technology, just as PET-CT I just showed you.
Like the biomarkers.
Yes, the biomarkers, the PET-CT. But before that, something like 10 or 20 years ago, even nowadays, in some underdevelopment areas, the diagnosis of AD, diagnosis of neurodegenerative diseases, still requires lumbar puncture. Yes, that is a very painful, invasive, and dangerous procedure. Despite it's regular, it's still dangerous and invasive. So we take the cerebrospinal fluid from the patients, and we detect certain biomarkers such as A beta, such as tau in it. And if there is a positive result, or if there is a certain pathological relevant pattern of those biomarkers, we say that we have most likely an AD or a certain type of neurodegenerative diseases. This is not ideal, as anyone can say that. If you have a patient which is senile, it's very difficult for you to always do this repetitive lumbar punctures.
Of course.
So a trend in today's medical society is we use biomarkers from peripheral blood. Peripheral blood, that means blood from finger. You pick a finger, you pick a blood, and now you have this. We have many other test tubes, even the pregnancy test, for this kind of thing. So you can, the ideal scenario would be one day in the future, of course, we have many preliminary kits for this kind of thing. One day in the future, you can just walk in the pharmacy and you ask for a kit, and you go home, you pick a finger, you take a blood, and now you know if you have the risk for AD or not. If you have that, you can start the treatment as soon as possible.
Okay.
Being said, I would like to say that there is many studies in the biomarkers for diagnosing neurodegenerative diseases using peripheral blood or peripheral plasma. And one of the most accurate biomarkers related to behavior, related to all the scoring system for diagnosing AD is tau.
It's tau.
It's tau. It's a kind of an isoform of tau. It's tau P217 or another isoform, tau P181. The relevance, the significance between those tau numbers and also the scoring is somewhere, I think the p-value was several zeros, 5.
Very relevant.
So it's very, very accurate.
That's right.
And that's actually another evidence telling us how tau is tightly connected to the disease, to the pathology, to the cognitive impairment.
Wow, good to know. Now we're providing the best tau model to the industry.
Yes, and actually you can imagine with these tau models, now more and more this kind of peripheral blood test kit can be explored, can be formed, can be investigated, can be invented using our mouse models. So you can take, it's not so easy, let's just face it, it's not so easy to collect blood from human patients. But under a complete strict laboratory environment and under certain ethical improvement, it is much easier for scientists to use mouse models. Now we can easily collect the blood from those mouse models to develop our own, our better kit for peripheral blood diagnosis for AD, for tau-related diseases.
Wow. So it's not just therapeutics, but also diagnosis. Yes, yes.
Diagnosis is always one of the most important things in neurodegenerative diseases.
Of course.
As you understand that there is a saying that every 10 years you are new you. because most of the organs inside you, the skins, the blood vessels, some of our bones, and some of our soft tissues, they regenerate, they recycle for a certain period of time, such as every 28 days we have new skins. It does not apply to tattoo, by the way.
No.
But there are two exemptions. One is heart. You have only one heart. You use that for life.
Brain.
One is the brain. Of course, there are cumulative evidence saying that there are neurogenesis in the brains, even in the adult brains. But the neurogenesis process is slow. So most of the neurons we get from the beginning of our life will be accompanying us by the end of that.
Yeah, of course.
So if neurodegeneration really happens, if neurons start dying, there is no way back. There is no way back. At least to the extent of our current knowledge, there is no way back.
So we need to
intervene the process as early as possible.
Yes, yes.
As early as possible. That's why the diagnosis is so important. And that's why we think our model can be a very powerful tool for developing new diagnosis method.
Wow, didn't know that before.
We have human tau. We have human tau sequence.
Yes.
That's why it's powerful.
Good. So this time we have this model. The full human tau sequence is working happily in mice, and it's showing us a good tau pathology, recapitulate the human disease, not just AD, but all tau-related
disease.
Yeah.
And it releases the tau as expected if everything goes well to the blood for all sorts of diagnosis process. Yes. This also goes for the treatment side.
Yes.
Well, the tau antibody treatment or any tau treatment is not as famous or as popular as A-beta treatment, right?
Yeah.
But there are some. So if for people to intervene the tau-related pathology, especially to intervene the pathology-related tau, the phosphorylation, the aggregation, you have to have the right tau. One of the most dangerous things for targeting tau is tau. The function of tau inside neuron is vital, it's important. So if you target the wrong tau, you create an artificial dementia instead of curing it.
Yeah, the tau itself is the skeleton inside the neuron.
Yes.
If you take it out, it's going to collapse. So
that means if we would like to develop a tau targeting treatment using animal models, we better be sure that the tau is recapitulated accurately in these animal models.
Yes, yes.
Which is ours.
Oh, wow. Great. So yeah, I hope that we continue to do research on this model and continue to provide this model to the community, research community, drug development community. And as I said, AD is too important to be ignored, even though it's the most important nut to crack, right? But we have to do it.
We have to do it.
Yeah, I mean, I guess most of us would see at least one of your close friends, close family members affected by AD. So it's to me, it's also very personal. So I would like to see that we can make a little bit of contribution. And
I would not underestimate this contribution that much. It's important.
It is important. Yes, yes. Let's continue to do our work.
Just putting myself back. 5 years ago, when I was still a PhD student, I would be much appreciated if I had this model. Yes.
Okay.
There are so many things we can do about it. There are so many research we can do about it.
Wow. Okay. So we've talked for close to an hour, I guess, and very happy to have this opportunity to talk to you on the most important disease model, and maybe next time we'll be on something else.
The feeling is mutual. I'm really happy to talk about it, and I'm really happy to present this kind of data, these precious models, to the scientific community and also for the industrial community for developing the tau-related drugs.
Yes, yes.
And yes, hopefully. Well, since the tau is, at least we partially completed our current study, I'm also working on other humanized models, focusing on neurological disease. Yes, we actually have several other humanized models. We have the humanized SNCA, which is alpha-synuclein.
Yes.
It represents another very important neurodegenerative diseases, Parkinson's disease, Lewy body disease, Lewy body dementia. And hopefully we are going to get the first round of result from those strain in the next few months.
Yes.
And meanwhile, we didn't stop our, let's just say, the AD mouse model construction, as despite we have mentioned that there are already several APP or A-beta related models.
Yeah, I heard we humanized the APP.
Yes.
Not just the full genomic sequence, but also the human promoter. So altogether, it's over 350 kb.
Even a little bit longer. A little bit longer, yes. And this time we are going to have another APP model, which is unlike any APP model we have had before. This time we have the entire human sequence in the mouse, and I'm very excited to see
the
what
HUGO
is
APP.
going to,
Maybe we'll talk about that next time when you have the results.
Yes. I can't wait to see the result, actually.
Okay.
I'm so curious about that.
Thank you so much, Morris. And so happy to have another episode with you, and hopefully we can do another one next time.
Next month.
Thank you very much.
Thank you very much.